Oh, I'm sorry. Thank you. Good afternoon, everyone. Thank you for the introduction. Let me just share my screen. And here we go. So what I'm going to talk about is this reclaiming energy sources through gas production evaluation of municipal solid waste, a mathematical model. The study was carried out in the city of Nogales, which is located in the northwest of Mexico, on the borderline to Arizona state. And the good management of solid waste is crucial since we can see a lot of problems derived from final confinement of this waste, and they have a social, economical, and environmental impacts. The regular confinement of urban solids in landfills is a challenge to municipal authorities, not only for the spatial issues that may come out, but the potential risks in terms of public health and pollution spread. It's been reported at solid waste generation per capita in Nogales by one organization of the government here as 0.72 kilograms per day for 2011. From all this, 46.5% corresponds to organic solid waste. The problem the final confinement of this solid waste in landfills poses is the production of leachates and biogas. The use of bioreactors to treat the organic solid waste permits to gain on an energy source, biogas, in lower time than the ones needed in landfills. This is an advantage. Besides, the gas collection and control help to diminish pollutant emissions to the atmosphere. To manage rubbish pile-up, a well-founded approach implies to treat these urban solids in a reactor so as to accelerate its decomposition through emulation of the biological process. It has been proved successful the digestion of organic urban solids to produce natural gas efficiently. Table 1 shows the composition of biogas, and we can see that this kind of composition can, give it some properties as a good, very good and efficient fuel. The objective of this study was to aim to model the gas productivity of the organic fraction of municipal waste as a function of time in a batch system with a mathematical equation. Materials and methods. First of all, we needed to determine the solid content analysis, and the samples were taken from different sources. 5 grams of solid waste was weighed to determine total solids from meats, cellulosic debris, vegetables, and a blend of all of them. For analysis of humidity, the samples were put into an oven heated to 105 degrees for 24 hours until dry weights were constant. This according to the Mexican normative. All samples were in duplicate. The total solid content was determined after taking the samples to constant dry weight at 550 degrees. This determination was very important to derive the whole study afterwards. The experimental design consisted in having the different samples arranged in Erlenmeyer flask, 1 liter capacity. The amount of sample depended on the total solid content calculated, right? All the flasks were identified properly and the samples were inoculated with a sludge from the local wastewater treatment plant in a concentration of 1 gram in 1 liter. Polyethylene flasks containing a brine solution, 30% mass in volume, were connected to Erlenmeyer flasks previously mentioned. Figure 2 shows the system, and we can see the experiment set up with anaerobic biological digestion. Each of the samples contained in the flask, the glass Erlenmeyer flask, were connected to the polyethylene flask, which contained the salty solution. And this one also to an empty Erlenmeyer flask that received the solution transfer due to the gas generation. Of course, all the pipelines in this case were sealed with industrial glue so as to prevent leakage. The system was monitored and inspected so as to register volume decrease in sample flasks along with the transfer of salty solution each 2 hours for as long as it needed. The results were treated with a differential equation to find a model that fitted the gas production as a function of time. The equation establishes that the variation of mass with respect to time is equal to a constant K, which depends on the material tested, multiplied by the inverse of time, as we can see in equation 1. Table 2 shows the total solid content in the different samples, and we can observe for meats, vegetables, cellulosic debris, and a blend of all of them. The solution to equation 1, as 2 points of the curve are evaluated, permits to describe the gas production in the system. And we go to the equation 2, where we calculated using the equation for density to convert the volume to mass. That's why our equation 2 describes mass as a function of time, where these M1, M2 are 2 values of mass multiplied by the logarithm of time t between time 1 plus mass 1. And here we have also that VT was the accumulated volume at instant t. Therefore, also the mass as a function of t is the accumulated mass. A graphic for mass production of gas as function of time is presented in figure 3. As we can see from this figure, there is consistency in the equation proposed to describe the production of gas as a function of time by the sample regarding the blend of vegetables, cellulosic debris, and meats. Okay, so from this we can conclude that to manage municipal solid waste is a real challenge to any administration in the cities. It seems unlikely to diminish due to the increasing population in most of Latin American cities. And anaerobic biological digestion is a viable alternative to treat solid waste. It can sum up to reclaim an energy source for a sustainable plant. And the model obtained fitted a great deal for the experimental results. These results can help estimate the yield of gas production in terms of temperature, inoculate, concentration, and mass water rate to feed the reactor. Eventually, it represents a reasonable method to scale up for technical feasibility in the treatment of solid wastes to produce biogas. This adds up value to the model found, since it let us scale up and treat industrial quantities of solid waste, especially the organic portion. So we would like to thank collaborators, our undergraduate student, Mayra Elizabeth Reyes Gonzalez, who recently presented her examination to obtain the master's degree in our school, and also Rommel Fernando Benitez Paz, our teacher and professor of mathematics. Okay, thank you very much for your attention. Questions are welcome.
Thank you very much. And if I understood correctly, so from your model, we can predict the amount of the biogas that will be released on the landfill, so which we can extract and use as a fuel. Is it correct, or did I miss the point?
No, it's right. You can use this equation to estimate the gas production, of course, and instead of letting this solid waste to get confined in the landfills, we better propose to treat them in a bioreactor to have control of this gas production and to avoid these gas emissions to go to the atmosphere and pollute the environment, right?
So did you run your test on at one temperature, like ambient temperature, or did you check also different temperatures? Because I can imagine that temperature, of course, in these bioprocesses has a significant influence. It can accelerate, right, the behavior of bacteria that run this biodegradation, or it can stop them from producing. So did you check different temperatures? How does it look like?
Actually, it was carried out at the ambient temperature. And we only have a small, very slight variation from 22 to 26, which is the average temperature in the month of April here in Nogales.
And do you plan to extend this research? And if you plan to extend this research to what are the future steps?
It's all right. We propose to our next generation of students to produce a small bioreactor, a prototype, so that we can control not only temperature, but pressure and concentration of the inoculate at the beginning.
I think it will be also interesting to check like addition of different bacteria, right? Because they can different bacteria can accelerate the production of biogas, and yeah, it will be interesting to hear results of your next research. No more questions from my side. Thank you.